Hello, my name is Caleb Rosenthal, and this project is in collaboration with Dean Young and is a continuation of research conducted by Maddie Bigdy and Dr. Betsy Martinez-Voss on Maize Rhizosphere microbiomes. So in previous research, they had sought to understand whether Triuret would be a suitable slow-release fertilizer. And slow-release fertilizers reduce algal blooms and other environmental impacts in contrast to normal fertilizers. However, part of their research concluded that Triuret rapidly degraded to biuret due to microbes that were a part of the Maize Rhizosphere microbiome. And from this, both Dean and myself were interested in that, and then also the various impacts that fertilizer has on the variation or the makeup, the composition of the microbes in the microbiome. So from that, they had sent in several samples for 16S rRNA sequencing from the microbiome of this rhizosphere and the maize plant. And we utilized both CHIME2 and DNA subway for metagenomic and bioinformatic analysis. And from that, we had drawn several conclusions and found several results as well. So if you'll follow me right here. The first one was from the previous work, and from that they had found that 25% or more usage or concentration of Triuret actually resulted in stunted growth of the maize plant, whereas biuret usage at all resulted in inhibited growth whatsoever. And then secondly, they had an idea that if they were to reduce the bacteria that were in the soil by an autoclave, that they might find successful subsequent growth. In which they did. We see all of them are in growth right here. So from this, and with this in mind, Dean and myself analyzed both normal soil data and then combined data. So within the normal soil data, this is just the non-autoclave sample, and we don't see any clear clustering or any clear trends here. So nothing was significant. However, when we did look at the combined data, so this is both the normal soil data and the autoclaved data as well, we do see two clustering samples. So this one right here is the normal soil, whereas this one right here is the autoclave sample. And then from this, we were further curious and decided to look at the taxonomic breakdown or the abundance of the bacteria within each sample. And we did find two different conclusions from that as well. So from this, we had identified that Triuret degrades, as I was saying before, into biuret rapidly. Due to the microbes that were making up the microbiome, which was later fixed by the autoclaving conducted by Maddie Bigdy. And then along with this too, autoclaving the soil reduced the previously present bacteria as well as altered the composition of the community. And that's why we see the separate cluster here in green in comparison to this teal cluster group right here, which is the normal soil data. The change was likely to reduce the bacteria, as previously stated, and then also autoclaving affects the bacterial composition way more actually than just the various fertilizer type, which was one of the questions that Dean and I were both looking at. And we see that in comparison by comparing both this graph right here, this principal coordinate analysis plot, which just graphs variation or difference in samples, in comparison to this graph right here. So we don't see clustering here, but we see clustering here. And then lastly, while looking at that taxonomic breakdown, we identified two interesting things. There was an archaeal thromboarchaeota sample, which we'll see right here in the orange, if you follow the pointer. And then on the other hand, we'll see a Pastöseria bacteria, which is this pink ribbon right here. And interestingly enough, the thromboarchaeota is likely due to the thermophilic and ammonia oxidizing characteristics that it has, while as the Pastöseria bacteria actually aid in nitrogen cycling, which is also probably why it was found there in the normal soil data in contrast to the autoclave sample. So that was likely reduced in this autoclave section. And then from this, we were wondering how we could improve or what we could do next. And so we were interested in actually core microbiota, and then with that as well to kind of change around how the autoclave and natural soil concept was designed, and play with that to see if we can find different results or find other interesting things as well. But so back to the core microbiota, like humans, plants also have several microbiomes. They have a microbiome of the roots, They have microbiome in their stems and stuff like that. And so the stem region is called the caulosphere, and I think that would be something that we'd be interested in for the future. And then lastly, I just want to say thank you to our the USDA Ridgeway Forum Fund, Dr. Betsy Martinez-Voss, Bruce Nash from the DNA Learning Center, and Catherine Malady as well. Thank you.